plants functioning under environmental stresses. As probably you noticed, on those classes we are not working with the most common stresses like drought or salinity, or not only with those. We want to present you also effect of other environmental factors which are also affecting plants negatively. Our team, me and Dr. Popek, we are mostly working with air pollution. So with those classes, we would like to present you two gases. One is outdoor, one is indoor, which have a negative effect on plants' performance. We'll start with the gas you will find indoor. It's called formaldehyde. Formaldehyde you can find in every room, in every building you can imagine. It comes from new furniture, from cleaning agents, from different chemicals and plastics used in indoor environment. We'll not use pure formaldehyde. We'll use formalin, which you probably know. It's to preserve animals, insects, organs for a longer period of time. The experimental setup is very easy, but as you can see, we try to be very careful with this compound. Even if this is just formalin, not pure formaldehyde, it's still negative for our health. So especially when you breathe it, it starts to be dangerous. So we'll use 4 jars. In 2 of them we'll have pelargonium. This plant will represent sensitive species, so plants which don't like to take formaldehyde too much. And the second species which we will use is Ficus benjamina. This species is pretty tolerant to formaldehyde. So after a few days, you would like to see a negative effect, a clear negative effect of formaldehyde on pelargonium and rather healthy plants of Ficus benjamina. What I will do, I will place in one of the beakers in each species a piece of cotton or something like that. On this cotton I will pour a few drops, 3 to 4 drops of formaldehyde, in our case formalin. So just few drops, it's enough to affect our plants negatively. Formalin is there, so try to clean it, to close it quickly. Same with ficus. 1, 2, 3, done. I will also close it. The control same way. The setup is simple, as you can see. The beaker, water, not distilled water, just standard tap water. There's enough of oxygen in the jar. We also close the controls because we want to keep control in the same condition as we do for formaldehyde treated plants. The symptoms are not easy to see, to recognize. You can see it when you are close to the plant, but it's not so easy when you see photos or videos. The plants sometimes look exactly the same as controls, but they are soft, they look like a little bit rotten. Sometimes you can see dark dots or areas on the leaves, but it's not as obvious as in case of other gaseous pollutants. Okay, thank you. So you were presented to effect of indoor air pollution on plants. So now we will do outdoor. As an example, we will take sulfur dioxide. The sulfur we will take from these sulfur stripes. We'll just make them fire, and the smoke will be pure sulfur dioxide. We have to prepare a little bit our experiment. I will just present you the plants we will use. The same species as for formaldehyde. So as a sensitive plant, we'll use pelargonium. As a tolerant, we will take again ficus. Here, the biggest difference is that we'll have to use oil. With the oil, we'll have to cover the surface of water, like this one. The oil is important because the smoke or the sulfur dioxide can react with water, and the effect of this reaction is acid. And then we don't want to see the effect of acid on plant. We want to see the effect of sulfur dioxide on plant. I will add oil to all the experimental combinations, including control. We want to keep plants in the same experimental conditions. Also the order is here important. So as you notice, I started with the water. The beakers were already filled with water. Then I placed the plant in the water, and then I put it. I pour oil on the top of the water. Plant cannot go through the oil because then we can block the veins of plants, and most probably the water uptake won't be possible. The controls I will just close like that, and with the sulfur we'll have to play a little bit. I will use forceps matches, so we need to have I am not smoking, that's why. We'll need to have. Okay, that's.
Don't burn our lab.
No. If you want to be professional, it's difficult to do it properly. And also, I have the plastic gloves. I just burn it. I see the smoke. So I let it burn a little bit, and then I close the jar. I'm not sure if you can see it, but we can see it very clearly. There is more and more smoke inside. Nice difference between two jars. And the same with ficus. I hope I will do it faster and better. Oh. Okay, smoke. And also you can see clearly the differences. The plants, I think they need about we need about the week to see a nice effect of sulfur dioxide on plants. The hood is on constantly, so the sulfur dioxide will slowly leak from the jars and it will be taken by the hood. So the concentration of sulfur dioxide will decrease gradually in the jars. Now it's extremely high, but at the end of the experiment will be pretty low. We will be able to open the jars, and after the week you will see the results of the experiment. Most probably we'll decide with Dr. Popek. We'll ask you to do a similar experiment at your home with the plants you will choose as source of the sulfur. You will just take more than one match, a few matches. Very easy to do at home, and you can see the difference between different plant species and their tolerance to sulfur dioxide. Thank you. Indoor pollutant, as an example, we took formaldehyde. As you maybe remember, we use a few drops of formalin, which was dropped here on a cotton wool. The same two species as we use for sulfur dioxide. So we have Ficus benjamina, acid tolerant plant. This one was exposed to formaldehyde, and as you can see, no changes. The color is the same. The structure of the leaf is also not affected by the pollutant. Again, completely different story in case of pelargonium. Control looks good, the color is okay, the structure of the leaf as well. And here is the effect of formaldehyde. So the color is completely changed from green to brownish. The change in color for formaldehyde effect is not specific. Sometimes it can be changed, as in this case sometimes it's not changed. You can see the same color, you can notice the same color, but the structure is completely different. And the second thing you can see, and this is very specific, is the structure of the leaf, which is very soft. It's a bit like a jelly structure, and I don't even want to touch it with my gloves. You can see that there is something on a leaf which doesn't look pretty nice. That was only few drops of formalin, which is not 100% formaldehyde. I don't remember the composition of formalin, but already you can see such a drastic change. Again, as in case of sulfur dioxide, we can distinguish two clear responses: a tolerant species, resistant to this pollutant, and very sensitive one, which is pelargonium. Our lesson, practical lesson, is that please keep at home as much plants as possible. All the plants with strong, thick leaves with a thick wax layer, they are much better protected from organic pollutants than others, and also via gas exchange and stomatas. Most probably they also uptake formaldehyde from indoor air, cleaning your air, purifying your air, and making your life better and safer. Here we have results of our experiment where our plants were exposed to sulfur dioxide. First ficus, that's control, just to prove you that plants under control conditions they look pretty well. And this is plant which was exposed to sulfur. The smoke is gone, as you can see, and also in this case the plant looks pretty nice. We cannot recognize any negative effects of this gas. Completely different story is in case pelargonium. Control plant, pretty nice, nothing happened. And this is the plant which was exposed to sulfur. You can also compare those two pieces, and you can even see that more smoke was generated in ficus jar. Here this is less burnt, but the effect is obvious. That's completely dead plant, very soft, the green color is gone. Also you can see the effect of sulfur dioxide on oil, which is also denaturated. So pretty obvious result. What is our main goal to what we wanted to present you? That we have two types of plants. One is absolutely sensitive to this kind of pollutants, and another one can do pretty well. From practical point of view, it means that those species shouldn't be grown in places where emission of sulfur dioxide is very high. On the other hand, those are adapted to this kind of conditions.